viruses are very fascinating if you are interested in molecular biology because they do a lot of things that are kind of sinning if you think about it. Like they're here to be sinners in our world, and I think that that's very cool. That they just come in here and they do these things that pervert our very existence. So why don't we why don't we get started? Let's see. Huh. I guess this is my first slide. You love sinning? Of course you do. Amy is basically a virus. She lives to pervert existence. Um anyway, viruses, here's what they are. They are biological entities that may or may not predate life itself. They are found everywhere on Earth in every single ecosystem and, according to some, in outer space. They can infect any living organism of any of the domains of life. It doesn't matter who you are. You could be a plant, you could be an us, you could be a bacteria, you could be an archaea. All of those things have the potential to be infected by viruses. No one is safe. Um, these absolute feats of biology and chemistry uh, far outnumber the amount of predicted terrestrial organisms in existence by a factor of 9,000. So that's a lot. That's a lot. And if we're talking just the total amount of like viral particles, that might actually exceed the number of stars in our solar system. So these guys kind of pervert math in a way, if you think about it. There are a lot of them. They surround us. And they're some of the most fascinating creatures in existence, despite the fact that they're not actually creatures. Which brings me to the elephant in the room. Are viruses alive? No. They're not. Scientifically, biologically speaking, viruses are not alive. Philosophically speaking, it might be up for debate, but I'm not a philosopher, so I'm not gonna debate, and we're not gonna have a debate. I'm here to teach, and you're here to learn. So just sit back and let me kind of demonstrate to you how unalive viruses are. For something to even be considered alive, they have to be classified. They need to demonstrate most, if not all, of the 7 characteristics of life. So, firstly, they need to living organisms need to regulate themselves in some way. They need to do homeostasis. So, us humans can maintain homeostasis in a lot of different ways. Like our hypothalamus can detect our body temperature and regulate that accordingly. That's a form of homeostasis. We also need to regulate our concentration of iron and our concentration of ions in our body so that they stay within a healthy range. Um, and this doesn't happen automatically. It doesn't just happen. There needs to be a stimulus that indicates that there's a change that needs to be made. So a living thing responds to a stimulus. And it needs to be detected by the organism so that it can respond accordingly. Like a drop in body temperature would be considered a stimulus that would need a response. Light is a stimulus that plants respond to by physically growing toward the light source, called phototropism. Bacteria, for example, can sense and respond to a food stimulus by moving toward it. And then after they eat the food, they can metabolize it. They metabolize the compounds into metabolites in order to fuel themselves. We actually all do that, not just bacteria, but we do that too. Living things undergo metabolism as a way to break down nutrients for energy and for building other necessary molecules. And because we are multicellular, we have a ton of different compartments and specialized areas in our bodies that can do very specific steps of metabolism. Speaking of cells, having them is kind of required to be alive, since cells are the basic unit of life. That's kind of a biology buzz term. Cells are the basic unit of life. Mitochondria is the powerhouse of the cell. We've all probably heard that a ton of times. Living things are organized as either a bunch of different cells or a single cell. And no matter what, they perform the metabolic tasks that are required for their overall survival, as well as their growth. An organism that is metabolically building, that is in a state of anabolism, more than it is in a state of metabolically breaking down or catabolism, is growing. And the growth of an organism is both physical and adaptive. Organisms are constantly adapting to their environments and evolving to become more and more fit for life. This, of course, happens over a very, very extended period of time, and it involves evolutionary concepts as well as molecular ones, where genes promote survival in an organism in a specific environment. Where the genes that promote the survival of an organism in a specific environment will be selected for and passed down to future generations. So living things must have the ability to reproduce within a species and without any outside factors. They just need to be able to do it. This can be sexual reproduction or asexual reproduction. And those are both considered like acceptable forms of genetic transfer. Personally, I know that I exhibit all 7 of these characteristics. So do you. And so does every other living thing, your dog, your cat, your snake, every single one of them. So let's see how viruses stack up to this. Yeah, no, oof. All viruses do is evolve, which they have to do because our bodies possess a lot of antiviral defenses, naturally occurring antiviral defenses. So viruses absolutely must evolve they must evolve quickly in order to evade those defenses. Foreshadowing here, some viruses have a double-stranded RNA genome. And double-stranded RNA is, well, our body wants to snipe that immediately because it is very unnatural and just doesn't really occur anywhere in life. You just don't see double-stranded RNA. So those viruses needed to evolve in order to encapsulate themselves in proteins so that they could trick their way into our cells. So, sure, viruses evolve, but they don't metabolize, they don't do homeostasis, they don't grow, they don't respond to any stimuli because those are cellular processes. And viruses are not cells. They're just genetic material. They're just genomes. And they have one goal no matter what environment they're in. Replicate. Replication is the virus's version of reproduction. But because it needs to use machinery from a host cell, it's not considered reproduction. Because remember, reproduction, you gotta just do it. You gotta just be able to do it. And viruses need our help in order to reproduce. So we call it replication. It's a separate thing. And while they do encode some of their own proteins, they still generally need to use our polymerases in order to actually transcribe and translate those genes, as well as make copies of itself. So a viral genome is very small. It'll encode its own capsid proteins, maybe even some enzymes, and there will be specific genes that it will make in order to, for example, evade the host genome. But we have a way bigger genome. We have like I forget if it's million or billion. This is embarrassing. I think it's 3 million nucleotides, and we already encode every single thing that we need to do in order to survive. All of our cells already are able to make everything that they need. Both of these genomes, though, can make their own replication enzymes. They can make their own transcription enzymes, and they can make, like I said before, make proteins to evade our immune system. But again, viral genomes need help. Especially when it comes to translation, viral genomes don't make their own ribosomes. They have to use our ribosomes no matter what. So viruses, they're biological entities, but they are not alive. They're on the very edge of life, and they've certainly played a role in the evolution of living things, and maybe even in the emergence of life itself. Because, fun fact, most viruses have an RNA genome. And the reason I'm telling you this is because the leading idea on pre-life, so 3 to 4 billion years ago, before there was life on this planet, the leading idea is that we had an RNA world. We had these free nucleotides floating around, these free proteins floating around, and the main nucleic acid was not DNA, but RNA. Wow. Hey, Buddha boy, how you doing? We're just getting viral up in here. Talk about early life. So, motherfucker. So yeah, that's like the leading idea, is that we just had RNA before, and then eventually it was switched out for DNA because DNA is more stable. But it really makes you think, the fact that most viruses are RNA kind of makes me think that they're old. There are 3 main theories on how did viruses come to be. And the reason that we kind of know these things and the reason we make these hypotheses is because viruses are able to integrate themselves into our genome. And when that happens, they can then be passed down to multiple generations, acting as a sort of fossil. And we actually call that a molecular fossil. Okay, good. I'm glad it didn't give you more ads. All right, so here are some of our ideas, the leading ideas on how did viruses start existing. And the first one is the regressive hypothesis. So basically what this says is that, what if there was a little cell? And then that little cell became a parasite of this big cell? And then over time, what if the little cell sort of got so dependent on its host that it lost its ability to, for example, replicate its genome by itself? So the early versions of, we're pretty sure that the early versions of cells all kind of had to work together, and they all had their sort of like special jobs that they did. So it's very likely that there could have been some sort of parasitic relationship that developed between 2 different cells. Oh, I'm sorry your hand is injured. Aw. I hope you're able to like take some drugs, some anti-inflammatories or something. Help with the pain and make you heal faster. How'd you hurt your hand? You don't have to answer because your hand hurts, but I'm curious and I care. So yeah, it was a free for all back then because cells kind of, you know, as life is also coming into existence, cells are, you know, really depending on each other at this point in life. And there is a huge virus that infects amoeba that kind of lends credence to this hypothesis. The Mimi virus is so massive that we originally thought it was a bacteria, and it kind of changed the way that we categorize viruses. Like, that's how huge it was. And it seems to encode a lot of its own components, like more than other viruses. It's still considered a virus because it does require host proteins in order to replicate itself, but damn, this guy can do a lot on its own. Dude, that's what I did to my wrist. I got tendonitis. It takes so fucking long for that shit to go away. And they ended up giving me something called meloxicam, which is like a stronger version of ibuprofen, and that was really helpful. So honestly, I know you have stomach problems, so I would be careful, but if your stomach can take it, I would keep on that ibuprofen. It'll make you heal faster. But that really sucks. I'm really sorry. So yeah, Poliovirus. Kind of a recent discovery. Um, the other thing is chlamydia. So chlamydia is interesting because it's a bacteria, and it kind of also toes the line when we look at the definition of life, because despite it being a bacteria, it can only live with a host. Yet we still classify it as a bacteria. So this lends further credence to the idea that these pathogens may have arisen as the result of some sort of parasitic relationship between certain cells. Something happened, something made it so that both chlamydia, a bacteria, and the Poliovirus, a virus, lost certain genes that allowed them to reproduce on their own. So that's the regressive hypothesis. And of course, it's a hypothesis. So it's got some problems. Like, we don't have a really good model for what the intermediate organism is. Like, okay, we can think about a little cell goes into a big cell, but what exactly does that look like? We don't know. We don't really have any leading ideas about that. Another important point is that Poliovirus and chlamydia both have double-stranded DNA genomes. And I just said that most viruses are RNA. So if this is something that happened like during the emergence of life, it doesn't quite add up, because we're thinking that DNA came along a little later in that timeline. And yeah, so it doesn't explain how RNA would have come into existence exactly. But that's okay, because all we need to do is just think of some other ideas. Like the cellular origin hypothesis. This considers the beginning of cell, which occurred billions of years ago. So as life itself was beginning to happen, which this is actually the leading idea, lipid-lined bubbles from the early ocean captured stray nucleic acid, stray RNA. That's the leading idea. Bubbles. So, with the cellular origin hypothesis and all these protocells being made, which is the nucleic acid inside the bubble, what if some of that genetic material just escaped from that and then went off and did its own thing? Because that's kind of what viruses are. Viruses are just genetic material that escaped and did their own thing. So it is possible that during that time where we don't quite have life yet, but we have these sort of protocells forming, that genetic material escaped. And the fact that some proteins do code for some very similar or even the exact same proteins that living cells code for, that kind of lends credence to the fact that, like, okay, this is coming from the same thing. And yeah, it's an interesting idea, especially since we know that genes do have the ability to jump. Yes, that's right. Genes can jump, which brings me to a tangent about women in science. So, jumping genes, or transposons, were discovered by Barbara McClintock when she was studying maize. She was, I forget specifically what she was studying, but it was maize that had broken chromosomes. And she was noting the different colors, not only within the corn itself, but also within the leaves. And she determined that not only could genes be turned on and off, but these genes could actually jump around and move in the genome, which completely, this is the 40s, this is the 1940s where she is discovering this. So we know about genes and DNA, but we don't even have the structure of DNA yet. The structure of DNA comes in about 1953. So this subverts everything that we think about genetics at the time. To such an insane degree that when she brings her findings to a symposium in 1951, the room is silent. Like, silent. no one could believe what she was saying. And I find it hard to believe that her being a woman didn't play a little bit of a role in this, but the things that she was saying were legitimately bonkers at the time. Like, it was very well accepted that genes are in fixed positions on our chromosomes and that they do not move. So it wasn't until 1983 when someone else discovered transposons that she was given the Nobel Prize for her findings. So just to take a look, we know genes can be turned on and off. I've talked about that a lot in my epigenetics lectures, but transposons they're just genes. They're just parts of our DNA that can move. And they increase genetic variation. I don't think they do tend to do a lot, but if they insert themselves in a really important gene, it is possible for them to cause disease. But I think that that's kind of rare. So transposons, those exist. Those are a class of mobile genetic elements. And guess what? Viruses are also a class of mobile genetic elements. So this really, it's really making sense here that, you know, since both of these things can kind of bounce around, it makes sense that maybe viruses escaped and they, you know, they have this ability to incorporate themselves in our genome and all that jazz. So I think it's logical to consider that they arose by escaping from the genome of a more complex human being. Or complex being, I should say. Of course, it doesn't really explain the fact that viruses do have proteins of their own and they can make proteins of their own, whereas transposons are just genes. It's just DNA. They don't make proteins. They're just genes that move around. They don't have any of their own protein structure. So this hypothesis doesn't really cover what's up with that. So yeah, it's difficult to imagine that viruses fully predate cells. It kind of seems like they're very much intertwined, because how could they do their own thing without the help of other genes and therefore other proteins? In walks our final hypothesis for how viruses emerged, the coevolution hypothesis, which gives viruses and life the same birthday, which feels right to me. It feels right that the emergence of viruses coincides with the emergence of life. So this hypothesis is fully stating that 3 billion years ago, we have these free-floating amino acids, nucleic acids, and I guess polypeptides. And some of these guys are going to go into the bubble. They're going to acquire a cell membrane. And eventually that's what's going to become life. Those are going to become prokaryotes and eukaryotes. But what if not all of these get captured into these bubbles? Maybe these nucleic acids that are just so happen to not be acquiring a cell membrane, maybe this is what becomes viruses. So this would be happening at the same time, which I think I like this idea. I think this one is very fascinating. So then the idea would then be that those nucleic acids that are becoming viruses, and they're not, you know, they would have to then rely on the organisms that are being formed, like the cellular organisms that are being formed at the time, and then also for the subsequent billions of years. I think that this is very interesting. The existence of subviral agents really makes this look like a great idea. So viroids, I know, vocabulary. Viroids are subviral agents. They're not quite viruses that infect plants. And the reason they're not considered viruses is because they don't code for proteins. They're literally just RNA. This guy right here, this is a viroid. It's just, I think this is like a single-stranded RNA even that has these hairpin loops. So they're also kind of like the transposons. They're just genetic material, but they can interact with the host cell and with the proteins inside of a host cell in order to propagate itself. So there's that. That's one type of subviral agent. There's also the hepatitis D virus. Also, not technically a virus. We call it a virus, but it's actually a satellite. Because here's the thing, you can only get the disease hepatitis D if you have already been infected with hepatitis B. And that is because hepatitis D can't make its own protein coat, which is an important part of a virus that makes it infectious. It needs Hep B to be present already, and then it basically steals the protein coat from Hep B. So this picture right here, these rods here are the hepatitis D satellite, and then these more circular cells are either hepatitis B or it's the hepatitis B protein coat with hepatitis D inside of it. We can't distinguish between those two, but because the presence of Hep B is required, we call that a helper virus. And then the Hep D virus, because it can't do this on its own, we call that a satellite. So the existence of these subviral agents makes this coevolution hypothesis look pretty dang sexy. And I think it's great, but there's always a but. It kind of implies that there's like a common ancestor to viruses, and that's just not. I mean, that's what we have for ourselves, right? You know, we want to kind of like copy paste. Like with life itself, we have these ideas about us having common ancestors, but that's simply not how it works when it comes to viruses, because their mutation rates are really freaking high. So there's no way for us to pinpoint a common ancestor. Something that we can do with ourselves. Like, we all have genetic qualities. We all have genes that are in common with each other. A human gene that does something, there might be that exact same gene in a bird or in a bacteria, maybe. Like, there are all these conserved things, and we just don't see that in viruses. So yeah, they just don't show those types of patterns, which is why when we classify them, it has less to do with this idea of the common ancestor and more to do with specific traits. Viral taxonomy. It's literally insane. It's just insanity. We have a very hard time following like a doctrine and just keeping up with the evolution of viruses. So much so that in 2020 there was a complete overhaul of both taxonomy and nomenclature. I wonder what the happened in 2020 that made us want to do that. But that's when the ICTV, the International Committee on the Taxonomy of Viruses, decided to make some changes to their naming protocols. And they're probably still renaming them right now. By the end of this sentence, they will have voted for a new taxonomy on viruses. I swear to God. Right here is actually straight from the ICTV website. So in 1971 was when we first logged this poliovirus. And I didn't show, but there's a ton of different name changes that occur until we rely on enterovirus Coxsackie pol, which is not correct, actually. Now that I've done this is actually not even correct what they did here, but, hell yeah. But you can see here that the lineage is constantly getting updated, and the way that we categorize viruses has completely changed since the 70s. It's completely different now. And it's completely different from us, you know? We have dumb kids prefer candy over fried green spinach. That is the taxonomy of life, and it is based on a common ancestor. And I'm going to prove to you that us humans are distantly related to bacteria. So we're going to take a little field trip, all right? All right? We're going to take a little field trip to a super sexy place. And I'm going to show you. All right? Here we are in the tree of life. This is humans, Homo sapiens, our green spinach, our genus and species. We share a common ancestor with chimpanzees, which arose 6 million years ago. So out of all of the other living beings that are not each other, we are the most closely related to chimpanzees because of this common ancestor that arose 6 million years ago. But let's zoom out a bit because our family is Hominidae, which is the great apes. And we all share a common ancestor. All of us great apes, we share a common ancestor that existed up here 25 million years ago. That is when we split from the great apes, which is where we come from, and the old world monkeys, which is like those little tiny small trickery monkey guys that you see. Those are the old world monkeys. Now 65 million years ago, that is when we see the dawn of our order, which is primates. So all primates of all types have a common ancestor that existed 65 million years ago. We primates, what's so special about us, we have our graspity hands, we have our eyes that are forward facing, and we have big brain. And we have very big brain. And those are things that set us apart from rodents, which we split off from 75 million years ago. So our common ancestor with rodents lived 75 million years ago. If we're gonna keep going back because our kingdom, I got eyes in the speak for yourself. I got eyes in the back of my head. Then I don't know where you are on this tree. Oh my god. We're mammals, but we're not just any mammals. We have babies. We give birth to young that we first gestate inside of us. For a gestational period instead of laying an egg. So we are that would make us therian, like we're placental, but we're also therian mammals. That's what I just described. So that's where we break off from marsupials, who give birth to their young and then carry them outside. We also don't use egg. We're mammals, but we don't use egg like echidnas do. So our common ancestor with egg mammals existed 180 million years ago. So we're at mammals. We do still have, of course, we have a common ancestor with other avians and reptiles that arose 320 million years ago. Because even though we gestate and even though we give birth, egg-laying individuals, so reptiles and birds, are still considered amniotes. We still come into life in a sac of some sort. We also have 4 limbs, much like a bunch of birds, reptiles, and amphibians over here. So we are tetrapods. So the common ancestor to the 4-limbed people arose 340 million years ago. Uh-oh, this is gonna crash. This thing keeps crashing. That's okay. It's usually fine. Okay, tetrapods. We're good. So we have, even though my hand looks completely different from a bird wing, we call those homologous structures because the bones in my hand, they look pretty freaking similar and seem to have a common ancestor to the bones in the birds of a wing. So that's thanks to our tetrapod common ancestor that arose 340 million years ago. As mammals, we also happen to be vertebrates. We have a spinal column that protects our spinal cord, and we have a cranium to protect our brain. And because our spinal cord happens to be located on our backside, we got to go back because we're not just any vertebrate. We are chordates. We are in the phylum Chordata, literally because our spinal cord is in the back of us. That's how it is. So humans, like every single other organism that I've compared us to so far, they're animals, but they're not just any animals. We're deuterostomes, which means we come into the world asshole first. Literally, our buttholes form and then it tubes up to our mouth. Whereas some other peoples, like starfishes, they are mouth first and then they tube down to the butthole. Teacher, why is my spine all bad? Oh my god. Cause actually, Gray, you split off way too late or way too early, and you're a bony vertebrate, so you're all crangly. Deuterostomes. I do love that word. So Yeah, this is animals. Deuterostomes are like, they're like us animals. Asshole first. Uh Unlike worms, bugs, crabs, other fish, they are not deuterostomes. We also have bilateral symmetry. So the common ancestor for that arose 570 million years ago, and that's when um us animals with symmetry split off from animals that don't have symmetry, like jellyfish and shit. But our defining quality is that we're multicellular. And now for that, please don't crash again. We have to go 800 million years. 800 million years. You could do it. Here we go. 800 million years ago. Wowsies, gowsies. That is when the lineage that became multicellular animals split off from unicellular eukaryotes. So they're still eukaryotic. They have a nucleus just like we do. And they have membrane-bound organelles just like we do, but they're just one instead of multiple like we are. So that's 800 million years ago that we split off from that common ancestor with unicellular eukaryotes. But we're not done. Because eukaryotes, of course, are composed of cells, like I just said, with the membrane-bound organelles and the nucleus and shit. So that first arose 2.7 million years ago when something absolutely fucking insane happened. 2.7. We're going. We're passing all of these. All of these are eukaryotic, but unicellular. 2.7. So this is where eukaryotes go. But this is when the event happened 2.7 billion years ago. This you're still a single cell being, sadly. Oh no. That's okay. All of your friends, all of us your friends will make up your multicellularness. So this is when the absolutely insane crazy fucking thing happens where a fucking alpha proteobacteria with the ability to use oxygen, it got inside of an archaeon. It just went right inside of it. And then that bacteria eventually became the mitochondrion. And then the whole unit somehow got the ability to like make cytoskeletons and shit and like other organelles, and then eventually it gave rise to what is now known as all eukaryotes. That's how it happened. It went right inside of it. Right inside. And that's how we started. And then it was a couple billion years after that. Let's see. Plants. Ha. This is when the crazy thing happened, and then after that, when eukaryotes become a thing, this is when animals and fungi and protists all split off from plants. So plants got to exist way before we did, of course. So that's insane. That hurts my brain. So we basically like have the remnants of a very, very early bacteria inside of almost all of our cells. And then, yeah, I'll zoom out more. So archaea and eukaryotes, this is our last common ancestor with archaea. And then our last common ancestor with bacteria is just the first. Whatever the first. Why do fungus? Well, fungus, they split here. No. I think they're here. Yeah, they're also pretty early. They're still eukaryotes, though. But yeah, they're very interesting. It's like, okay, you're not fucking plants. What the fuck are you? They're very different from plants, apparently. But we're all connected. We're all connected on this tree of life. I did just go from humans to bacteria, and we're all just there. Fungi have gills? I did not know that. If that's true, I had no idea about that. But this is crazy, right? Our cellular makeup, our genetics, and sometimes our anatomy connect all of us. They connect all life. And they remind us that we probably came from the same place. Not viruses though. Not at all. Oh yeah, mushrooms. Well, mushrooms are fungi, but I don't think that those are gills in the same way that fish have gills. I thought they were different. I don't know. Yeah, mushrooms don't breathe water. That's why I don't. I think it's like a different kind of gills, but they do have gills. So viruses, I can't do this with viruses. That's impossible. They're not like this at all. They don't have dumb kids prefer candy over fried green spinach. No, no, no. They have, let's go back to our little thing here. Viruses have to be special. They have, Richard Kind probably can only feel good sometimes. That's what they have. It is literally such a shit show. There's no way I could do what I just did with humans. I could not do that. I couldn't take one virus and do that exact same thing. It's such a shit show. Because for a virus, they're kingdoms, they're phyla, species, whatever the they don't like show up at specific points in time. And they don't have traceable common ancestors generally. No. A committee has to study them and then vote on how viruses get categorized based on their shape, their host range, their genome, what diseases they cause, all that stuff. So we use this wannabe life classification system. Like, it's just kind of, we're trying to make it like look like our own. We use that one. And then we also use Baltimore classification, which is way easier because it's just a genome. Is it a double-stranded DNA virus? Okay, cool. Group 1. Way better. But we got to use them together, unfortunately. And you know what? I'm sick of labels. I'm just sick of them. Let's cleanse our palates with some virus basics. Let's get away from taxonomy and labels for a second. I'm sick of them. Mass spectraturians have RNA and they can eat food that humans eat because we have DNA. Very interesting. What? I love the idea of an RNA model for life because it's like a window into what we probably had 3 billion years ago. I love that idea. So let's just palate cleanser. Let's look at let's just look at the viruses, okay? Let's look at them. Let's look at how they enter our cells and replicate. Let's look at how they all interact. Wow, it didn't like Coom. That's interesting. They can't Coom. That is wow. I'm shedding the taxonomy for now. Viruses all interact with our cells and with our genomes, and sometimes that leads to infection. In a nutshell, this is basically what I'm gonna show you. This is basically what needs to happen in order for a virus to successfully replicate itself. So, number 1, it needs to attach to the cell membrane. And it does this by binding to a specific receptor on the cell membrane. We all just came off of COVID, so I'm sure you all know that the receptor for our favorite coronavirus is the ACE2 receptor. So pretty much all viruses have that. They have like a specific receptor that they like the most, and that determines the type of cell that they're gonna like the most. Like, for example, hepatitis viruses love liver cells, and it also determines the host range. So if there's a receptor that's in primates but not in like cows, then that virus can probably infect, apes and humans, but it can't infect cows, for example. So it determines the host specificity as well. And yeah, I already said the cell type. The next thing that has to happen, come on, is penetration. So after the virus enters the cell, then after it binds the receptor, it either comes in through endocytosis, which is when the cell eats the virus, or it comes in through
Yo, guys, I'm
membrane
enjoying a
fusion.
crisp Diet Cock.
Either of those two. That's
I mean Diet
penetration.
Cock. I mean Diet Cock. I mean Diet Cock.
Oh my god. Gray, if you can win the prize, you could do such a special Diet Cock spam command. I don't know. Thinking of ideas. Um After that, when it's inside of the cell, it's gonna uncoat. And so now you just have like these capsid proteins with the genome inside. And depending on the type of virus, it's either gonna do its thing in the cytoplasm or it's gonna go to the nucleus. And depending on where that is, it's gonna replicate. It's gonna make copies of its genome. And it's also gonna make some of its viral proteins. Because they need to both replicate their genomes to make a bunch of copies of themselves, and then they also need to replicate their genome so that they can do translation and make the proteins that they need. And again, this will look different depending on the type of genome, whether it's DNA or RNA and stuff like that, because a DNA virus will need to replicate its DNA, and it will then need to transcribe the DNA into an mRNA. Whereas a virus that's already RNA, it can probably just immediately do its thing without any of that fluff. I hate it when viruses dump their genetic load in my cells. I know, it's so rude. Like, pull out game week. Oh wow, we climbing. Alright, so after a bunch of copies of the genome are made, like I said, some are gonna go and they're gonna help make proteins, but some are gonna go and they're gonna be assembled into these capsids. And these guys are eventually gonna be the ones that are gonna go out from the cell and infect other cells, but they're not quite ready yet. Exon is a gas station. It's also a gene that codes for a protein, whereas an intron is junk DNA. So you just graduated from junk to useful. That sounds really bad. But that's what an intron and exon are. Oh my god. Alright, so after they're assembled into their packages, oh yeah, more vocab, I'm still sorry. So like, you have a virus, right? But what we have here, they're getting packed into virions. And a virion you can think of as the extracellular and like infectious form of a virus that occurs after it's assembled. And then the virus is like the intracellular form. And it also refers to the life cycle as a whole. So you're going to hear me say virion when I get to the assembly section, because they're being assembled into virions because eventually those virions are going to be released and they're going to go and infect other cells. And they can be released in a couple of ways. They can explode the cell in a bunch of virions will come out, and that's the lytic cycle. They can also bud across the membrane and like kind of slowly just murder the cell. And then we have this weird thing called the lysogenic life cycle, which is where a virus will stay dormant within a host cell, potentially also inside the host genome, and it will keep the cell alive as it has its replication cycles. So, this is basically what viruses are doing when they're infecting our cells. Obviously, it looks different depending on genome type, and we're going to see that, but this is more or less what is going on. They come in, they make a ton of themselves, and then they undergo this mass exodus so that they can find another cellular host for them to take advantage of. Isn't that what TB does? Yeah, I think TB does something similar, but it's a bacteria. So, it's slightly different than this because I think, rather than doing like genome replication, there's like a specific toxin that messes with your cell in a certain way. Like cholera does something like that too. So I'm not sure how similar it is, but I do know. But tuberculosis is a bacteria, so it might be slightly different. So Yeah, that being said, even though most viruses do this, they have their own little special ways of doing it, which is what makes them super hard to um categorize, but we do it anyway. Oh yeah, wait, I wanted to say um virions, when virions are being released from a cell, that's called viral shedding. And if your cells are actively shedding viral particles, you're contagious. That's considered, that's what being contagious is. Um, but yeah, so hard to categorize, even though they all do the same kind of thing like this, but we try to do it anyways. But first, there's a little disclaimer. I have no idea how to pronounce any of these upcoming words. I just simply have no idea, especially the different names that are used in the ICTV classification. These words are just really insane, and they make really feeble attempts at like cramming a ton of information into one word. They're impossible to pronounce, and you'll see what I mean, and I'm sorry. So first up, we have a very, very familiar face here, all right? If you try to say them, you'll summon a sex demon. Dude, yeah. Huengognivire. Negarnaviricota. They really... I'm speaking in tongues. Ricotta cheese. I feel like Pisonnaviricites is the one that summons the sex demon. And it has a piss fetish. Did you guys have the chickenpox? Did you guys ever have it? Cause I didn't, cause my mummy got me the vaccine. Hell yeah. Amy has a scar actually from the chickenpox. But it, the official name, hell yeah, we vaccinated here. The official name for the virus, this came after the new naming. So within the past 5 years, this was renamed to Varicellovirus human alpha 3. Wow, it rolls off the tongue. It's great. For the longest time, it was known as VZV, or the Varicella-zoster virus, because it also causes shingles or zoster. If you were a kid in the US getting your kindergarten shots after 1995, you probably got a varicella vaccine. And that's what I'm going to call it also. I'm calling this varicella. You know what I'm talking about now. Varicella is the Varicellovirus human alpha 3. Oh no, not one from the vaccine. Amy, I think, probably didn't get the vaccine. You are 2 years older. So it probably wasn't available. It was also, I think, a very controversial vaccine or something, because I think there were like lies about them using like aborted fetuses or something. I don't know, but a lot of people didn't want to get it. Is that the one that looks like a butthole closed? You mean her butthole? Where is the scar? It's on her arm. It's butthole-ish. So yeah, chickenpox is oh, it's for smallpox. Interesting. I probably got a smallpox vaccine. That's probably the ones that you get when you're born, maybe. Oh, it could be. Oh, yeah, you might have gotten like a completely, I could see that. I don't know if they do that anymore. But who knows, in Mexico they might have. Yeah. That'd be interesting. But for the most part, after 1995, people get injections of this, and it's not common. My mom is a preschool teacher. There's outbreaks every once in a while, but it's pretty rare nowadays because of that. And varicella has a double-stranded DNA genome, so it is in group 1 of the Baltimore classification, and this is reflected in its realm. Duplodnaviria or Duplodnaviria. I don't know which one it is, but it's one of those, and it means double DNA. So that's its realm that tells us that. And then it's trying to convey that it's double DNA. Like, I see what they're doing, but it's just too weird. The rest of it, the kingdom, phylum, whatever the heck, are assigned based on its structure as well as its host range. This is a human-specific virus. Only humans get Varicellovirus human alpha 3. Only humans. And the order, though, we're going up. Herpes viralis. Now, this can infect mammals and reptiles, amphibians, birds, and shellfish. So, viruses in that order are pretty prolific. But then the family, Orthoherpes viridae, that also has the herpes simplex virus. And a clear commonality between this virus and herpes simplex is the existence of the blistering. Structure-wise, herpes viruses have a special matrix of protein between the capsid and the envelope called the tegument. This is specific to herpes viruses. And the structure is needed for like viral replication. It's required for that. And it also helps the virus get into the nucleus of the cell, which is where we need to do our replication because it's a DNA virus. And it also helps assemble the new viral particles, as well as help evade the immune system. So it does a lot. It does a lot. And remember, it's just herpes viruses, interestingly, that has this tegument. So the virus can spread through respiratory droplets, or it can spread through like open blister fluid. And then when it comes in contact with epithelial mucosa, especially in the upper respiratory tract, that's where it's going to start replicating. So when, let's say, someone coughed in your mouth and they have chickenpox or whatever, or they rub their blisters on you, blister fluid. Let's say they rub their blister fluid on you. The first thing that's going to happen is attachment. The virus is going to attach to the cell membrane, probably through one of these two receptors, and then it's going to fuse itself to the cell membrane and release it into the cytoplasm. So you can see here, the envelope goes away, and now we just have the capsid with the double-stranded DNA in it. Rub their blister! Chickenpox party! I know, the blister fluid. Oh my god. Oh my god. So yeah, now we have the capsid with the genetic material. That is then gonna dock at the nucleus, and it's gonna inject the double-stranded DNA into the nucleus. And then this is where the magic is gonna happen, because first, the immediate early genes are going to be expressed, and these guys help with the transcription of the early genes, and they also still help with immune evasion. So viruses will do a lot of things to evade the immune system. And then the early genes are going to get transcribed, and these are the ones that are going to help with replication. And they get transcribed by our RNA polymerase. And so these proteins are going to help with replication. They're going to help replicate the DNA. Now, DNA polymerases, whether it's the viral one or if it's ours, they can't just make DNA from nothing. You can't just, like, even if you have a template, you can't just, boom, DNA. They need a primer in order to do that, and a primer is just a short string of RNA nucleotides that will need to be present on the template so that the polymerase will have a place to start. I've covered this in other lectures before, but basically DNA has a handedness to it, where you have a strand that ends with a 5' phosphate, and then you have another strand that ends with a 3' phosphate. So one goes 5 to 3, and the other goes 3 to 5. That's just the handedness of it. So not only can DNA polymerase not just make DNA out of nothing, but it has to add nucleotides to the 3' hydroxyl. It absolutely has to. It cannot add to the 5' end. Gattaca. You're getting ranks just by being here. I set this up a while ago, and it's weird. It sometimes it doesn't do anything, and sometimes it does. So it's working now. So you went from being just a little gene to now you're a little prokaryote, and maybe you have a tail and everything. um. So what do we do then? If the DNA polymerase can't extend the DNA with the template, it can't just make DNA out of nothing. What do we do? Well, isn't Varicellovirus so smart? Enter primase. It encodes its own mother effing primase, which puts down a primer, this green thing here, so that DNA polymerase can bind and extend it. If there were no primase, there would be no DNA replication. And Varicella makes its own primase. It doesn't need to use any of ours. It just makes it by itself. So now, with the existence of this primase, which is an early gene, it can now replicate the genome to its frickin' heart's content. After enough particles have been replicated, the late proteins come in, and these guys are gonna make the parts of the like the protein coat that the genome is gonna be assembled into. So that would be the capsid, the tegument, and the envelope. And this is what's needed to assemble the final virion. So here we see the the DNA that's been replicated. It is being packed into these capsids within the nucleus. It is then budding across the nucleus, and it is maturing and obtaining more proteins, and then it is getting released from the cell and going on to be its own infectious self. It goes on, and it can go and invade other cells. So it starts usually in like the respiratory tract, but then the virus will spread to the tonsils and other lymphoid tissues, where they will gain access to our T cells. Now, replication and release from T cells means that the virions can be carried in our bloodstream, and therefore eventually carried to our skin, which is where the skin that's what causes the skin lesions. It's because the virus is infecting T cells. The T cells are bringing it to our skin, and then it is causing blisters on the skin. And lesions for chickenpox, these can happen all over the body, all over the face and body. It does not discriminate. But of course, you can get chickenpox twice. So what's happening there is, after the infectious stage where you get chickenpox and stuff, varicella can still lie dormant within our sensory ganglia, so within like specific nerve cells. It gets carried along the axons of our nerves to like other sites within our nervous system where it'll just kind of chill. So it's difficult to study this. This is kind of like basically what's going on, but because Varicellovirus human alpha 3 only infects humans, it's hard to make a model, of what this looks like. Like, we can't just use rats for this because the virus is not going to infect rats. So this is basically what we think is going on. We don't know exactly where it becomes latent or like what happens during this latency phase. We just know that it hangs in some of our neurons as an episome, which is like a little piece of DNA that's separate from our and just chills out. And then reactivation happens due to stress or age or being immunocompromised. And once that happens, then wherever the virus decided to embed itself, that's where the rash is going to be reflected. So if the virus, let's say the virus took root in the right side of your C4 spinal nerve, you're going to have a rash probably either in the front of your like upper chest or your upper back, because it's going to be reflected in that dermatome, which is an area on your skin that is innervated by a specific nerve. So the dermatome for C4 is here, and it's also down here. You could probably get it on your arms too. dermatome. So notice how this is very different from chickenpox, because chickenpox just is everywhere, but because of the varicella lying dormant for an extended period of time in a specific nerve, zoster or shingles is only going to happen in that specific area. It's not going to be all over your whole entire body. And in case you're wondering, like I was when someone that I knew had shingles and hugged me, if you've never had shingles, or if you've never had chickenpox, and someone who actively has shingles and has blisters touches you, you can get chickenpox from that. So don't let any of your old ass relatives who have fucking shingles and are like, give me a hug, don't let them tell you that you can't get chickenpox from it, because you can. I'm hearing and reading this slide in Spanish. We don't have a word like chickenpox for it. We just call it varicella. La vacuna de varicella. Do you say varicella or do you just say varicella? Yeah, that's awesome. That's because that's more like accurate to what it is. It's just varicella. So that's really cool. Yeah, so you want to, oh, I love that. Yeah, you want to be careful, because I feel like I think my parents ended up getting like vaccinated for chickenpox. Or no, my mom had chickenpox when she was 21 years old, and she almost failed out of college because of it. It was really, really bad. So she was very insistent on like, it's 1998, all my kids are getting chickenpox shots. But yeah, don't let your relatives try to be like, don't worry, it's fine. It is not fine. If you've never had, and I've only had one varicella vaccine. I don't think I got a booster or anything. So yeah, be careful. Now, all this talk about varicella and herpes viralis and whatnot, you might have herpes on your mind during this discussion. Now, remember that varicella and herpes simplex are in the same family, and herpes simplex can also lie dormant in our nerves. Specifically, herpes simplex virus 1, HSV1, really likes to hide in your trigeminal nerve, which is in your face. And that HSV1 is the one that causes cold sores. HSV2 really likes to hide in your sacral ganglia, which of course innervates your genitals. So HSV2 is generally what causes genital herpes. So Varicellovirus has been a really wonderful example of a double-stranded DNA virus that enters our cells via membrane fusion. It expresses genes in specific phases, and it demonstrates latent infection a la herpes zoster. Yeah. You know I couldn't just put a dick on there. I had to do it to him. I had to do it to him. Oh my god. So, yeah, the more you know. Viruses. Staying in noibs. Another virus that we've 1,000,000,000% come in contact with is the rotavirus. It is the leading cause of gastroenteritis in children worldwide. So if there was like a vomiting bug that went around in your elementary school, it was probably a rotavirus that did that. There's a closely related virus called the norovirus, which causes the winter vomiting bug. So it's one of those. Yeah. Yeah. But colloquially, we call this the stomach flu, even though it has nothing to do with the influenza virus. It's not actually the flu. Is herpes dangerous? I think comparatively, it is not dangerous. I think it can be a danger if you are, immunocompromised. So I'm thinking of like, let's say that I had genital herpes and I wasn't open about it, and I had unprotected sex with someone who was like immunocompromised. I feel like that could be like a potential dangerous situation, but I think it's more annoying than dangerous. Oh, you had rotavirus? Oh, that sounds so terrible. Oh my god. Yeah, this little bugger really gets into your shit. And because it mainly attacks enterocytes, I feel like it can really, that those things are connected to your brain and stuff like that. So that's some messed up shit. But it causes a stomach flu, and it's not a flu. That's just a colloquial thing. It has a double-stranded RNA genome, so that's in group 3 of the Baltimore classification, and its realm is Ribovaria. This means that it uses something called an RNA-dependent RNA polymerase. That's all it means. The kingdom tells us that it has an RNA genome. Its phylum Duplornaviricoda, Duplo RNA viricoda, or whatever, shows that it specifically has a double-stranded RNA genome. Add the order, the family, the genus, and we get a virus that is unenveloped, icosahedral in shape, and genetically segmented. The rotavirus. Oh yeah, feverish state. Oh my god. That sounds terrible. Yeah, being an adult and getting rotavirus is very ouch, I imagine. It's a weird guy. I mean, if we look inside this capsid here, I said it was segmented. So there are 11 different double-stranded RNA segments in here. So they're apart. They're in segments. They're not together. So each gene has its own strand of double-stranded RNA. It's kind of like a chromosome in a way, a little bit. Rotavirus likes to invade the cells of the upper GI tract. It likes those enterocytes. And so that's why it causes really, really severe gastrointestinal symptoms. What was the other thing I wanted to point out? No, that's fine. So like varicella, it attaches to the membrane, but instead of fusing, it gets in through endocytosis. The cell eats it and it packs it into a vesicle, called an endosome while it's inside the cell. The surface proteins, here, VP7. Which one? VP7 and VP4. VP4, I don't know why it's not put here, but it's VP8 plus VP5. That's V4. That's the spike protein. So the spike protein and VP7, sorry, they mess with the membrane of the endosome. Now, endosomes usually have like a ton of calcium inside of them compared to the cytoplasm. But the rotavirus causes the endosome to spit out calcium. And once the concentration of calcium inside the endosome is more or less the same as the cytoplasm, then the endosome will break and then the rotavirus will burst out and it'll leave behind its top layer of proteins. So then you'll have this right here. So no more top layer. Just this guy. Just this little particle right here. It's really, really important that the genome stays inside of this particle because it's a double-stranded RNA, which remember is really unnatural. Our cell does not want to see that. So replication happens within this coat right here. And here it is. The new mRNA, these little green things coming out. This is the new mRNA that is eking out of these little pores here while the double-stranded RNA just stays where it is. Yeah, I hope they did too. I'm sure it did. I'm sure you got that frickin' immunity for life from that motherfucker now. Oh yeah. So the double-stranded RNA stays in here while the mRNA just kind of ekes out of it. And again, if we compare this to varicella, this is all happening in the cytoplasm. Whereas because varicella is a DNA virus, all of its like replication and stuff happened in the nucleus. So, and another thing that's different is at no point are we ever going to have a completely uncoded virus. It is always going to eke out its little mRNAs through these pores because it doesn't want to reveal the weirdness that's inside of it and potentially get killed by the cell. So, the mRNA that's coming out of this capsid, it's not only going to get sent for translation to the viral proteins, but it is also going to get packed into the proteins that are being actively translated from the other mRNAs up here. So now the newly coded mRNA particles here can replicate themselves so that they are double-stranded once again. And this is like I said, they all have to be single-stranded when they're being packed in. But then once they're in here, in order for it to actually be the fully formed virion, it's got to go back to being double-stranded RNA. And this is a better depiction here. So we've seen this already, where the virus, it comes in, it gets transcribed, blah, 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 translation. But this guy goes in and it gets packed, and then the double-stranded RNA synthesis happens. Ooh, okay, I don't want to mess this up. Rotavirus is how they filmed Waking Life. And a scanner darkly.
Hi.
Rotavirus time lapse of it duplicating, but I punch it in the face. Hi, Chrissy.
Yo, guys, I'm enjoying a crisp Diet Cock. I mean Diet Cock. I mean Diet Cock. I mean Diet Cock. I mean Diet Cock.
Okay, I wanted to specify because this is important. Hope you're doing well, friend. All of these mRNAs coming out of here are positive strand because the plus strand is what's correctly going to be translated, which means that you need to you're making minus strands from this as a template. And because the RDRP, the RNA-dependent RNA polymerase, has to add nucleotides from the 3' end, it's always going to use the positive one, as a template. Wait, that's actually not right. I'm alive, that's what matters. I feel that. We keep going. You know what? Nope. It's fine. We're not gonna, I'm thinking too much about it. All these little guys are going to be used as templates to make double-stranded RNA. And because this is a double-stranded RNA virus, it has to do that in order for it to eventually leave and become a virion. So what happens right before it does that is it picks up the rest of its proteins that have been made from translation, and then it leaves by lysing the cell, by killing the cell. A bunch of them do. And also this particle right here, it's not depicted here. Oh, maybe it's depicted here. No, it's not. This right here can also go back here. Like if I just put an arrow from here to here, it can also be used to make more mRNA for transcription. And you're going to see that in a lot of viruses where at a certain point, it can either go to be the virion or go to make more copies of itself to make proteins. So once it's assembled, this is when it is able to leave the cell and go on its merry way. So really quick, we just looked at two viruses that have a lot in common, and yet they have two completely different genomes. We have double-stranded DNA for varicella and double-stranded RNA for rotavirus. They both enter the cell through some receptor-mediated process, but varicella actually fuses with the membrane in order to enter, whereas rotavirus gets in through endocytosis and is thus encapsulated by an endosome, which is good. It wants extra protection. Varicella, a DNA virus, must go to the nucleus in order to take advantage of our DNA replication machinery. But rotavirus doesn't need any of that because it's already RNA. It doesn't need to be transcribed ever. So it stays partially encapsulated in the cytoplasm, where it makes copies of positive sense RNA that can then be packed or translated. Varicella will also replicate its genome all at once because it's just one big old thing and then pack it into the nucleus, whereas rotavirus is segmented. So each of its 11 genes will be replicated separately because they are separate double strands of RNA. So they both, with an asterisk on varicella, kill the cell and release many virus particles. For varicella, its realm is what categorized it as double-stranded DNA, which was Duplonaviria. But for rotavirus, we needed to get to the phylum before we knew that it was double-stranded RNA. Meh already, but this next one is really gonna throw a wrench in our comfort with viral taxonomy. It's gonna make things a little more messed up, I would say. Poliovirus causes polio. And it's a disease that we don't see very much of nowadays because of eradication efforts. But it is not fully eradicated. So with this guy, we're still in ribaviria because we use an RDRP. We're still in RNA virus, and then the family specifies this specifically a positive sense, a single-stranded RNA virus. And the rest just specifies the types of proteins it has. It's non-enveloped, it's unsegmented, and the transmission involves our intestines. Hence, enterovirus. The species Enterovirus C simply wasn't enough to categorize the poliovirus. So we have the different serotypes. And it's based off of like little differences in these capsid proteins. And because they're serotypes, this is referring to the immune response that they bring and like their antigen proteins basically. This means that immunity to one of the serotypes does not mean immunity to the other ones. Now, of the 3 serotypes, PV2, poliovirus 2, was eradicated in 2015. 3 was eradicated as of 2012, and then 1 is not fully eradicated. It still makes the rounds in Pakistan and Afghanistan. But yeah, so the fact that not all of them are eradicated at once kind of demonstrates how the antigen proteins are a little different. It is very small. It's considered one of the smallest or one of the simplest viruses out there. I mean, just look at it. It frickin' just pops itself in there into the cell using the CD155 receptor. And because it's a positive sense RNA virus, but it's just mRNA at that point. It doesn't need to go to the nucleus or anything. It's just right there. I read a book about polio and it scared me shitless. Oh shit. It is fecal oral, yes. Eat the poo poo. Or just from poor hygiene kind of thing. But anyway, yes, it's a positive sense single-stranded RNA. So that's mRNA. So it doesn't need to do any fancy things. It can just come into the cell and boom, start being translated by ribosomes pretty frickin' immediately. So it gets to doing that. It makes the necessary proteins that it needs to make. And before replication, it needs to be put into a vesicle because replication involves a double-stranded RNA intermediate, which our cell hates. We hate double-stranded RNA. So it makes the vesicle, it puts a copy of the positive sense RNA in here, and then it replicates it by making a double-stranded RNA intermediate here. Do I have a no? Some of the new we can continue making the positive sense mRNA from this intermediate. So some of that will be made and go and just be translated continually. And then some of it will get pushed into a capsid in order to form a virion. because it's an enterovirus, poliovirus will start in the intestines and like replicate and stuff. But we've all seen how it can end up causing paralysis. So at some point, it does take root in the motor neurons and okay, I know which positive sense single-stranded RNA virus you actually want to hear about. So why don't we just jump over to Her Majesty herself? The best-known member of beta coronavirus pandemicum. Yes, that is the name that they gave it. When do you think they gave it that name? Pandemicum. I'm mad. Anyway, the best-known member of beta coronavirus pandemicum, SARS-CoV-2, the virus that causes COVID. Note that until we get to the order, it is the same as polio. But Nidovirales is an order that specifies that it's a lot bigger than the poliovirus, and it is enveloped. So coronaviruses will steal from our plasma membrane to get some of their proteins, like varicella does. And then the rest just denotes specifics about their structure. And then beta coronaviruses are grouped together because they infect mammals. Bats are mammals, and they tend to be reservoirs for beta coronaviruses. We're not sure where this virus came from exactly, but there was clearly some zoonotic event. Zoonosis is the process of sickness coming from animal to human. There was clearly some zoonotic event where it hopped from another mammalian species to us, and it was very likely bats. The horseshoe bat specifically. There's a bat coronavirus that is within beta coronavirus pandemicum, and it is very, very similar to the one that caused the pandemic, like eerily similar. And nice. Oh, she hasn't seen Pride and Prejudice, the one with Keira Knightley. Oh, that's gonna be fun. I love that movie. It's so good. I hope you have fun. I'm gonna have so much fun talking about this fucking coronavirus. Oh my gosh. Yeah, that's gonna be great. And this is pretty much it, by the way. Nothing weird happened. There was no weird things. This kind of stuff, this zoonotic events are bound to happen as us humans continue to encroach upon the homes of other species. So, this is just what's gonna happen. Now, you may be wondering, strain? Why strain? What happened to serotype? Like I said, we're still figuring viruses out, so it's clunky. A serotype refers to the surface proteins that our immune system responds to. So infection with one serotype of like the rhinovirus, for example, which causes the common cold, it won't protect you from the other many serotypes of that virus. So that's why you could just get a cold all the time because of these gosh dang serotypes. A variant is based on genetic differences in the virus that distinguish it from other variants, but it's still considered that virus. It might not change the antigenic properties, so it might not be a different serotype. These are the variants of SARS-CoV-2, and they differ genetically, particularly in regards to that frickin' spike protein that we love so much. And they might also have different rates of like transmissibility or mortality, or they might respond differently to different drugs. But they are still SARS-CoV-2. There is only one strain of SARS-CoV-2. There's just the one. But there are multiple strains of beta coronavirus pandemicum, like SARS-CoV-1 would be another strain. SARS-CoV-1 is the virus that caused the 2004 SARS outbreak. So we can say that all strains are variants, but not all variants are strains. Thank you for coming to my vocab lesson. Anyway, back to real virology. So beta coronavirus pandemicum it's a positive sense RNA virus. What does that mean? It means low freaking effort. It means that the genome is inherently infectious, like the poliovirus. So it waltzes into the cell already primed to be translated. Oh yeah, let's go to Viral Zone to look at its structure and replication. Viral Zone is a really sick ass freaking website that I've been using. So let's get there. Okay. So this is the virion of SARS well, this is beta coronaviruses in general. Well, basically this is SARS-CoV-2. This is the spike protein right here, and this is the RNA that's been packed in protein. And you can see whenever I let's make this smaller. If I highlight something on the virion, it is going to highlight it in the genome to show me where it's encoded, and vice versa. So that's kind of cool. We have this. This is a spike protein. We got the M protein right here. It's a really tiny protein. The nucleoproteins here that pack it are right here. So that's really cool. So the spike protein is the one that's going to bind to that ACE2 receptor and gain entry. So let's look at here's how it happens. So here she is, knocking at our chamber door, binding ACE2, and then it can either come in via endocytosis or fusion. She does it both. If this TMPRSS2 receptor is also present with the ACE2, it'll fuse. So that's kind of cool. She can do both. So she comes in endocytosis. She's got an endosome. And then much like we just saw for polio, the endosome is gonna release the genetic material into the cytoplasm, and the cytoplasm or the RNA is gonna uncode, because right now it's curled, it's wrapped in protein, so we undo it, and then we can start translating. So you can see right here that some of the proteins that SARS-CoV-2 makes, they do some messed up things in our cells. It shuts off translation, and it makes it so that other proteins can't be trafficked within the endoplasmic reticulum, because it wants all of the focus, all of the resources to go into itself. It's a very, very selfish virus. Then what we have here is so this is translation of the proteins right here. They're being put on the ER membranes. It can also go and be and this is still in the cytoplasm. It gets replicated, and then we have these copies of the viral genomes here that get wrapped up into the N protein, and then they get packaged into, now we're in the Golgi. The Golgi is where proteins are packaged up, so it makes sense it's here. It's getting packed with the other proteins. And then we have, now it's a full virion, and boom, it can just go. So that's really cool and very similar to polio and not varicella, to polio and rotavirus. These RNA products will continue to be replicated and translated just continuously. Now, that's all well and good. This all makes sense, but what is going on over here? Subgenomic RNAs. What in the world are those? Well, let's go back here. Let's look at the genome. So this is another diabolical move by beta coronaviruses, where they can create these smaller RNAs that code for even more specific proteins. So if we go right here, these are the subgenomic RNAs. So this one just has E, this one just has M, this one just has N, this one just has 6, NSP6 or whatever. And this is fascinating. So there's something about the genome itself that is allowing the RDRP, which is doing the transcribing, to jump around to different areas of the genome, and allow it to only transcribe one little part of it. thus making multiple of these little subgenomic RNAs from the one viral mRNA particle. So these guys will go on to make proteins that will sabotage the cell as well. So, for example, we have these proteins over here that are going to make it so that nothing else can go inside the nucleus. This one is going to inhibit interferon signaling. Interferon is a communication molecule that's like an antiviral defense. So it shuts off a cell's antiviral defense. Some of these guys will also go and mess up MHC. So that's major histocompatibility complex. It's going to mess up our immune system's ability to like recognize what's going on. And it is going to prevent the cell from using its autophagy is something that happens when your cell is really stressed out, where it like breaks certain things down and reallocates resources. So beta coronaviruses will also prevent that from happening. So all in all, this virus really just ruins everything. It really, really does. It's truly evil. This is probably happening in the respiratory tract, probably the nasopharynx. That's where all this stuff usually starts. Reset. I don't know if it does like a full reset, but it's tackling it from a lot of different areas. So if they are inhibiting interferon signaling, then they're specifically inhibiting the signaling that says, hey, there's a virus, we gotta f this up. So it stops that completely. Oh. And then it also, the MHC disruption, it just prevents regular immune cells that might not be specific to a specific infection. It prevents them from recognizing that something's wrong. And then it's also, I mean, it's just preventing translation and trafficking. So anything else that your cell might do between those things that could ward off a virus, it's just not equipped anymore. Yeah. Yeah, this is a mean guy. This is a very mean guy. Okay, so we gotta stop that guy. So the main way that the virus spreads, of course, is through respiratory droplets. So it tends to start replicating in the nasopharynx. And ACE2 has decent expression there. That's this line right here. But as you can see, ACE2 and the other receptor that we saw there, the TMPR SS2, they have a lot more expression in the GI tract. Viruses really, really seem to like it there. You can get reinfected with SARS-CoV-2. I personally got it twice, 2 years apart, and it's really hard to define like what is reinfection and what is just prolonged infection, because viral shedding of these guys can go on for a really, really long time. But basically, it's defined as a positive test more than 90 days apart after an initial positive test. And that's what likely means that you got reinfected by another variant. These newer, better variants tend to be more adept at evading immune defenses. So you may be wondering why we don't call these different variants that may evade our immune system serotypes. And to that I say, great freaking question. You know what's really fucked up about that too is it didn't say that you had to have a negative result within that time. So it's trash. All the data on a lot of this is such trash. And the guy or whoever wrote this like paper, they were like, yeah, so we define it as you have to have a positive test more than 90 days after an initial one, but there's nothing that says that you have to have a negative test. And it's like, but you should probably, because then that would indicate that you're not infected anymore, and then you got reinfected. It hurts me. You know what we're going to do now? Is we're going to go to the flip side. Flip side. To negative sense single-stranded RNA viruses, which is group 5 of the Baltimore group. Influenza A is our example. It causes the flu, the real flu, not the stomach flu. And is its phylum Negerniviricota is what specifies it is a negative sense RNA virus. It is also segmented, as we know from its class and order, and it infects humans and some other mammals and maybe some birds. These viruses are very fascinating for a couple of reasons, but let me first show you what's weird about their replication process. So it starts like any other virus sometimes, and it binds its receptor. In this case, it's sialic acid. And it does that in order to get taken up by endocytosis. Then the endosome will eventually deposit the single-stranded RNA into the cytoplasm, along with some proteins. And then the proteins in the RNA will get transported to the nucleus. Normal so far, except not really, because none of the other RNA viruses that we talked about ever touch the nucleus, ever. And this is because the influenza virus actually doesn't have the ability to make its own mRNA, like the rotavirus and SARS-CoV-2 and the poliovirus did. It needs to use our machinery for that. And it also needs to steal nucleotides from us to make its mRNA. So all that needs to happen in the nucleus. Literally it steals nucleotides from our own mRNAs, and it does that through something called cap snatching. This is really cool. So these are viral proteins right here, PA, PB2, and PB1. These are all this is a protein complex that is completely coded for by the Influenza A virus. And it is going to right now it's bound to its own genome. It's single-stranded, but it's in a loop. And it's gonna bind our mRNA, the cellular mRNA, and it is going to cut off right here. It's gonna cut off the 5' cap from this cellular mRNA. So it's gonna cut it off, and then it's gonna put it opposite the 3' end. Excuse me. It's just gonna put the hat on its viral mRNA. It's viral RNA, and then it's gonna. Now with this new 5' cap, it can transcribe it. You don't need to listen to anything up here, but it's going to transcribe the rest of it. And then, now the viral RNA, guess what? You need a 5' cap in order to be recognized by a ribosome anyway. So this is like super smart and super evil that it does this. I think this is so cool. That literally evil genius behavior. So that's how it helps make its copies that can be packaged or translated. And the new mRNAs with their stolen caps will leave the nucleus to go get translated by ribosomes. This green thing I'm guessing is a ribosome. And two of those proteins that get translated, hemagglutinin and neuraminidase, are going to be sent to the cell membrane. Remember that. The others will go into the nucleus where they will form the capsid. So the capsid assembly, unlike other viruses, this is actually happening within the nucleus. So that's also very interesting. And then it will bud from the nucleus. And then as it's budding from the cell membrane, is going to acquire that hemagglutinin and neuraminidase. And this all happens very fast. These viral particles, these flu particles, can be detected like an hour after initial infection. That is super, super fast. And you may have noticed that hemagglutinin and neuraminidase, they start with H and N, respectively. And that the subtypes of Influenza A utilize those letters. Very astute. The H and the N, like in H1N1, they directly refer to these two proteins. There are 18 known H's and 11 known N's for us to mix and match with. Why do we call these subtypes and not serotypes? Since they seem to qualify as antigenically different? Well, that's because technically a serotype is a more general term, and that's that. That's the explanation. That's the reason. That's the only reason. So we have a ton of subtypes of this virus. We have so many. And as an RNA virus, it does tend to have a high mutation rate. So this accounts for its infectivity and our need to potentially get flu shots every flu season. But there's something else that really increases this virus's ability to cause pandemics. And that's reassortment. So Influenza A has 8 different segments to it. Like this is the segment that has the hemagglutinin, and this is the segment that has the neuraminidase, and then these are all the other segments with all the different genes. Remember that am I wrong? Is it varicella? Varicella, yes, was also segmented. So it's like 1 strand per gene kind of thing. This, in addition to the high mutation rates, Influenza A viruses, they, in addition to the high mutation rates, having more than one Influenza A virus infect a cell at the same time can lead to this process of reassortment. So if you have one subtype that's, open and it's actively replicating its genome, and then you got another one who's also replicating its genome, then what can happen is, parts of each of their genomes can get packed into one capsid. And now we potentially have another subtype. This is one subtype. This is another subtype. This is a brand new subtype that came from accidental mixing or reassorting of these two guys. The swine flu pandemic of 2009 involved H1N1, which was apparently a mix of swine, avian, and human influenza A virus genes. Like hybridizes like an oak, baby. So that's why the flu seems to be this ever-present disease that we never seem to be fully immune to despite vaccination. Speaking of immunity, HIV is a virus that tanks your immune system by using a process that completely reverses the central dogma of molecular biology. Speaking of perversions, my God, it is still in riboviria because it encodes an RDRP. But it also codes for an enzyme called reverse transcriptase, which uses an RNA template to make DNA. That's reverse transcription. Once that double-stranded DNA has been made, once the double-stranded DNA has been made, so here it is right here, it has circularized, it gets taken into the nucleus, and then it gets literally integrated into our genome using an enzyme called integrase. And it gets inserted into just a random part of our genome, completely random. At this point, it is called a provirus while it's within our genome. That's like any time a viral genome is inserted into a host genome, that's called a provirus. And HIV's favorite cell to infect are helper T cells. Its surface protein just interacts really, really well with those cells. I'm actually going to show you all a video that will better represent what HIV is doing when it infects cells. So let's go to it. Let's take a look at how HIV infects our cells. All right, here we go. So this is the virus, and inside we have the capsid proteins, and we have two strands of RNA, and then reverse transcriptase at the very, very ends here. So it's going to go to a helper T cell, and it's going to directly fuse with the membrane, but first it's going to attach to these CD4 receptors. And then it's going to attach additionally to another chemokine receptor at the same time. And then binding to both of these receptors, this is really cool. It's going to kind of like reel it down and bring it closer to the membrane until it fully fuses to it. So that puts the genetic material in. Both layers of the capsid are going to be degraded, and then these other proteins are going to go to the nucleus. This is reverse transcriptase right here. So the purple is it's making one strand of DNA, and then it is also going to take that strand of DNA and it is going to make it double-stranded. This is all happening in the cytoplasm. Once that happens, integrase is going to bind to it, and it's going to bring it into the nucleus. Ba-ba-boom. It's going to find a random area of our genome, and it's going to make a nick, and it's going to insert it into the genome. Then RNA polymerase can make an mRNA out of that gene. The mRNA will go back to the cytoplasm where it's going to be translated by a ribosome. So this is going to be a membrane protein right here. It's going to be packaged and sent to the membrane where the other ones are. These are the ones that were there from the initial fusion, but it's going to send more of them to the cell membrane. More strands are going to be translated into other proteins. So these right here are eventually going to make like the capsid protein that's going to hold the 2 strands of RNA in the virion. So this one, it's traveling. RNA, reverse transcriptase, and proteins are traveling together. They are going to get assembled, but it's not ready yet. First, these have to be cleaved by protease so that they can actually become like the specific parts of the capsid protein. And then, boom, there's the 2 RNA right there. And then they can bud off and continuously make copies of themselves because they're integrated. Yeah, that shit's really cool. Yeah. People always do like a really, really good job with the animations there. Okay. So, yeah, that's different from what we've been seeing. We've never seen reverse transcription where we take RNA and make DNA. That's very interesting. That's happening in the cytoplasm. And then it is being directly integrated into our genome with integrase. And then it can just continuously make copies of itself. And that's why it is so evil, because it's keeping the cell, the cell is not dying, so it is just continually making copies of itself. This is like the true lysogenic cycle, basically. And then, yeah, these are just some of the transmission routes. So the top, blood transfusion, if you get a blood transfusion from someone with HIV, you're gonna probably get HIV. Mother to child, it's different depending on like where it happens, but I think it's like 25%. You could get it during gestation. It can happen during birth, and it can also be transmitted through breast milk. Needle sharing is significantly lower than those top 2, as is sexual intercourse. There's a lot of stigma surrounding HIV. It so much that it literally used to be called GRID, or gay-related immunodeficiency. But such a big question. Where did this virus come from? Did it come from monkeys? Did it come from the gays? Did it come from a Canadian flight attendant? Did it come from drug addicts running rampant? I think there's only one right answer to this when it comes to just how this lentivirus got to humans. Colonialism, obviously. This is like partially like a real thing and partially my own opinion, but so there's this recent like common ancestor to one of the HIV groups, and it arose in 1910, which coincides with a lot of colonialism in Africa. I want to say specifically West Africa. And what do you get with colonialism? You get the spreading of cities and the spreading of everyone's legs, because prostitution becomes like really, really popular during this time. And by extension, venereal diseases are very, very common. They're all the rage in the early 1900s. And it's well known that having an existing STI can increase our chances of getting an HIV infection. It predisposes you. So what else is happening during colonization? More people means more hunting, more bushmeat practices, more moving into areas that were previously peacefully inhabited by animals, namely monkeys. And monkeys can get simian immunodeficiency virus. It's different from HIV, obviously, but similar enough, and it can pass to humans, but it is weak and it doesn't really do much. And it passes to humans not through sexual intercourse, by the way. It just exposure to like people who are like hunting the animals. No one's having sex with monkeys. I want that to be clear. But it can pass to humans and it doesn't really do anything. But we think that it doesn't tend to even transmit human to human, but because there's so many more people living together and the population is like increasing in these areas due to colonialism, there's more people to come in contact with. So hypothetically, that SIV could jump from human to human and then mutate into HIV. So that was my TED Talk on where HIV came from. There are the way that we treat HIV is by kind of developing multiple drugs that tackle the virus at multiple points in its replication cycle. There are drugs that prevent the virus from even coming into the cell. There are ones that inhibit reverse transcriptase, and it either does that by literally binding to the enzyme and stopping it, or by preventing the addition of nucleosides to the DNA. Nucleosides are just like the bases, basically. It prevents it from doing its job, but specifically by making it so that it can't add bases. Some of the drugs will prevent the DNA from being integrated into the host genome, and others are protease inhibitors, excuse me, which can stop the virus from maturing. So it breaks down those proteins that are meant to like form the final protein so that they can't ever be together and you can't make virions. Since HIV doesn't have a proofreading mechanism, it doesn't have a way, like when we like replicate our DNA, we have proofreading that helps prevent mistakes and mutations. Reverse transcriptase does not have that, so it can make a lot of mistakes leading to mutations that could potentially become resistant to some of these medications. That's why HIV positive people tend to do a combination therapy. They don't just take one of these things, they'll take multiple. All right. We are on my last virus example, which is hepatitis B. Really cool guy. It comes from a family of enveloped small viruses that affect the liver specifically. Hep B also utilizes reverse transcriptase. So it is in group 7, but it is a double-stranded DNA virus that transcribes into RNA, and then that RNA gets reverse transcribed back into DNA before it becomes a virion. The replication cycle of this virus is like nothing we have seen before. So, like HIV, it can be spread through childbirth, blood contact, sexual intercourse, and needle sharing. It can also be shared by pierced body jewelry. I did not know that. So you can get hepatitis B from sharing an ear piercing with someone who has it. So if any of those things happen, the virus will enter the cell by endocytosis, likely involving a receptor that is mainly found in the liver. We're not sure which one, I don't think. Yeah, like any body piercings, that's how, I know, yeah, it's, I feel like I would assume that it would have to be a new piercing because it's not healed all the way, but there was no, like, real specificity on that. So that's kind of scary. Um, Sorry. Once it's been endocytosed, the capsid with the genetic material gets released into the cytoplasm, and then that capsid and genetic material will be trafficked to the nucleus, where the genetic material will be released. The DNA will be released in here. Since and the reason it comes to the nucleus, of course, is because it does need to make wait. Yeah, because it needs to be transcribed from DNA. That's why we can't just do this in the cytoplasm. We gotta go to the nucleus. And at this point, the form of DNA that it's in is called rcDNA, which is relaxed circular DNA. You can kind of see it here. It's mostly double-stranded, but it has an area of single-strandedness. So it's not completely circular. It is relaxed circular DNA. And actually, you can better see it right here. The ladder here is double-stranded, and then here in these areas, this is single-stranded. So once it enters the nucleus, our host enzymes will ligate it. It will complete the circle so that we have our covalently closed circular DNA. So it's like completely circular at this point. It's all zipped up. Let's look at how this weird genome gets transcribed because it's really, really freaking cool. So here we have our closed, it's been ligated already by our stuff. It's come in and it's been completed by our host enzymes. So our host RNA polymerase is going to go on the outside on this orange strand right here. The orange strand is the minus DNA strand. And it is going to transcribe in a circle, and it's gonna over transcribe. It's gonna go past the point where it would need to stop, so that when you have it in a line like this, you have a second DR1 region. So we call this pgRNA. This is pregenomic RNA. So once it's transcribed like that, it leaves the nucleus, and then the P protein, which is the virus's own reverse transcriptase, it's gonna bind to this specific site right here, this little loop, this epsilon site, sorry. And it's gonna transcribe a little bit in this direction, because you have to go 5' to 3'. A little bit this way past the DR1. Then what's gonna freaking happen is it's gonna jump over here to the start of the 3' end. And it's gonna base pair just fine because what did this is just basically the same as what it just transcribed over here. So that's why we have that little, or that's why it has that little end hanging off there. So it jumps over here, and now it can fully replicate from that pregenomic RNA template. And while that's happening, the reverse transcriptase is gonna degrade this so that there's basically none of it left. Only this DR1 part. And then this part is going to jump over here and base pair with, it's the same as over here. It's going to base pair right here, and then it's going to transcribe. Again, you got to go 5' to 3', and this is circular. So it's going mute, and then this is just the continuation of the circle. So it just looks like this. And then you have, once again, your relaxed circular DNA. This is very cool. This, and it's this loop right here, this epsilon site, that lets the virus know that, oh, this is the pgRNA that I'm gonna like replicate right now. Very cool. So this is the form that is gonna be put into virions. Yeah, so that's what's gonna be going into virions, which is over here. So the pregenomic RNA, it's packed in the capsid. This is where everything that I just said is going to happen, like right here. So it can either go on to be a virion and infect cells, or it can go back to the nucleus where it can get continuously replicated. So if the Hep B virus is continually like, if the Hep B virus is continually making these longer pregenomic RNAs, then how do we get these smaller ones right here that eventually translate into the important proteins of the virus? Well, the genome has multiple start sites. So there are different areas where our polymerases can start transcription, leading to these differently sized transcripts that will code for different proteins. I believe also that the long pgRNA will be packed, reverse transcribed, and then either, like I said, recycled or further processed with proteins that become the infected virion. I haven't talked about it much before because it makes the most sense to talk about it now, but other viruses are able to find different start sites here and just make different mRNAs based on where they're starting in the genome. And then there's also this thing called leaky scanning, which is where a polymerase can like intentionally skip something. It's very cool, and it's very fascinating. Over here, you can see that some of these proteins are coalescing to form these empty guys called SVPs, or subviral particles. They don't have any genetic material in them, and they exist in order to further trick our immune system. They're basically decoys. Also, something that I mentioned at the very beginning was that the Hep D virus is a satellite that requires previous Hep B infection in order to cause disease. And that's because Hep D is stealing these viral envelopes from these subviral particles. So that's Liddy. This is very fascinating to me, the DNA to RNA to DNA. It's like, why? But also I get it. Alright, so this is the last thing I'm going to talk about is frickin' vaccines. These are injections of various types that help us become immune to certain diseases, some more deadly than others. I'm mainly going to talk about the ones that involve viruses, so like I'm not going to talk about the toxoid vaccine, because that's for, bacteria and shit. But yeah, these are important. The measles vaccine was made in 1963, and you can see that there was a steep drop in measles cases after that. The same with polio. The polio vaccine was made in 1955, and again, pretty steep decline after its development. To this day, at least in the US, it is required, asterisk, for us to get rounds of vaccinations, particularly when we're born, and when we first start going to school. Not all vaccinations are the same. There are different types that demonstrate different approaches, or that demonstrate different approaches to developing a vaccine. The entire pathogen can be used, or just parts of them, like specific protein subunits can be used, or just the genetic material can be used to make a vaccine, as we see with our favorite mRNA vaccine. So first up, inactivated vaccines. This is where the pathogen is killed or inactive. So when injected, the antigen, the dead antigen will get taken up by our cells. This will start the process of like our cells presenting the antigen to other immune cells, making antibodies, and then committing the antigen to immunological memory. It is, so there's you're going to see there's two parts to adaptive immunity. There's cell mediated and then antibody mediated. Inactivated, really, it's a little bit of cellular, but it's mostly just antibody mediated. And some of the pros are that it can't revert to a virulent form, which we kind of can see with some of the live attenuated viruses. And because it's relatively weak, it's okay usually for people who are immunocompromised. The cons, because it's weak, it likely requires boosters, and they're also very, very slow to make because they need to be cultured in a lab. The injectable polio vaccine is an inactivated virus, as it are most of the influenza vaccines. Next, we have those live attenuated. So the virus is alive, it's live, but it's also kind of nerfed in some way so that it's not infectious. When injected, it will activate both the cellular and humoral or antibody responses, which is pretty litty. This will lead to both T and B cell activation, so you will have IgM production, which is like your antibody immunity, and then you'll also have that humoral immunity, which involves like cells besides antibodies. These are great because they last longer than other vaccines, and they're pretty quick to make you immune to something. You also don't need to take boosters, and they're cost effective. However, there's this thing that can happen where the virus can revert to its infectious wild type if there are not if there isn't sufficient vaccination within a population. Pregnant people and immunocompromised also should not take live attenuated vaccines. Some examples: MMR, we all get that as a kindergarten shot, I think. The oral polio vaccine, which I feel like I watched a documentary a while ago of a little boy who got partial polio from it because, again, it's a live attenuated, so there's the potential for it to revert to its wild type. Smallpox, which was the first vaccine ever made, was live attenuated. And then varicella and shingles and rotavirus are also live vaccines. Subunit vaccines, they're only using parts of the pathogen that are needed for an immune response, so they're not, that's what makes them different from like live or like an infectious particle. It's just like a little part of it. And so the subunits, depending on what they are, they'll elicit some type of immune response, and then you'll get immunological memory from that. The types of subunits used are generally like a protein, so like maybe a part of the capsid of a virus. There's also no reversion to the wild type. It's pretty safe for immunocompromised, but again, it's weaker than the live ones, so you would need boosters. And apparently it's not, if you have like allergic reactions, it can, they're known for causing like certain allergic reactions. The Hep B vaccine is a subunit vaccine, so is Gardasil, and there's another shingles vaccine that's a subunit vaccine. So these are all, using the whole virus for the inactivated and the live, and then using parts for the subunit. We got genome vaccines. mRNA vaccines use a copy of viral mRNA that encodes some antigen that will cause an immune response. So it's an engineered copy of viral mRNA. So the mRNA, when it's injected, it'll get taken up by cells. The cells will then translate that mRNA into an antigen that will then stimulate adaptive immune responses. And there are hypothetically ways to engineer it so that you can lower innate immune activation. So you can lower, like, sometimes you get sick after you get a shot. You can maybe lower those side effects. And you can also maybe increase the half-life so that your body has more of a chance to replicate or translate it. So the pros are that it, unlike the killed vaccine, it also stimulates cellular immunity a little better, so you have a more robust immunological memory. And it does not integrate into the host genome. The cons are that it has to be stored. It has to be like refrigerated in very specific ways, so it could be hard to transport. And then there's also potentially some issues with autoimmune problems or immunocompromised people. So the Pfizer and Moderna vaccines, the COVID vaccines, these were the first and currently, I believe, the only mRNA vaccines. mRNA vaccines have been in development for a few decades, but it wasn't until this pandemic that production really sped up. And, like, the technology just had to be kind of like, right now. And I think that it's really great, the idea of having your own cells make an antigen and being able to, like, engineer it in a way that it's not integrating into our genome and it's not really, like, dangerous per se. And then lastly, we have DNA vaccines, which are also, their genome genetically engineered. They're plasmids, so they're like circular DNA that have specific sequences that encode antigens. But what's different is than the mRNAs, they get transfected into our cells. They get, like, integrated into our cells, and then they would have to be transcribed into mRNA and then translated. But these are theoretical. There's no DNA vaccine on the market right now. There might be some in development, like people attempting to do it, but right now they just don't, they do not exist. So, oh my god, my voice is actually dying. I've never had my voice be dead by the end of a lecture. I'm usually okay. So, yeah, those are the, I feel like there's this thought that DNA vaccines would have really low, really high immunity and really low side effects, so that's why they're in development, but right now the other ones are all we've got. And yeah, viruses are very, very cool and scary. And the fact that, like, we have developed these ways of hindering them, very cool. Very awesome. Heck yeah. Thanks for listening and hanging, guys. It was a grand old time. A fantastique time. I'll get my sketty out soon. Sketty schedule. And we'll have a great time. Round of applause for Amy. She's here. You are sleepy. Yeah, let's get going. Thanks again, friends. I'll hopefully see y'all later in the week for some litty times.